Thanks so much. Very much appreciated, and I'm super keen and super happy and super grateful to be here and so presenting on our work. I'm hoping that everyone can see me and can hear me. So if someone can give me a heads up that that you can see the slides as well. So and then we can then start. So. I guess to put things in context, up until, and I'm going to go back to this, I'm going to go back to this in a second. So up until a couple of years ago, so most of the work that I've been doing together with, at least with some of the colleagues in the community, was focused on, let's say, computational phenotyping for rare diseases, but predominantly in the space that is pre-diagnosis, or at least trying to get to some sort of an answer into diagnosis and sort of working on for those of you that are part of the rare disease community or are sort of knowledgeable in it, on diagnostic odyssey that is usually the journey that the patients go through in order to get an answer. So a couple of years ago, we started looking into perhaps a part of that community of rare disease patients, which is that as a community is undiagnosed, because about 40% of the rare disease patients do not actually have a diagnosis. And the question there is not necessarily uh, trying to get to an answer as much as how do you manage the condition, and how do you place I guess sort of the right interventions at the right time without actually having a label for what the condition is. So to take a step back from that, and I guess to split the problem into smaller problems and look at it from a scientific perspective. I guess the critical aspect that was missing from what we've seen was the idea of time and understanding how time plays in general in the phenotyping space in rare diseases, and in particular, what are the relationships that one can I guess can infer or can build across time between phenotypes. So that was kind of, I guess, where this has started, and what the work that I'm going to present today is, I guess, a first step in that direction. We were not far from being there, but I guess it's a first step in that direction by basically verticalizing the problem into a single context of a single disease, which was Noonan syndrome, and simplifying it a bit by just looking at the onset of the phenotypes, and I guess just focusing on, let's say, on a more static view of I guess of time, which is just the onset of phenotypes, without necessarily still looking immediately at relations, but with the hope that this first step then would help us or other perhaps that are interested in this field to take that next step. So I guess although I'm assuming that probably everyone I guess in the audience is sort of aware of this. I thought I'm just gonna throw in two slides, so that I shouldn't make any assumptions on, I guess, the foundational sort of components of this work, as well as of most of the work that has been done in the rare disease community over the last 10 years, which is one being on the human phenotype ontology, which is the de facto standard for representing phenotypes in humans. And it's ontology that at this point in time has about 17,000 concepts, has grown significantly over the last couple of years and over the last 5 years or so. And organized in principle. If you look at the top levels, the top level abnormalities by the anatomical structure or anatomical entity that's being impacted, as well as by anatomical type, as you can see, and going from a more abstract to more specific, from higher in the hierarchy to lower in the hierarchy, to something more concepts are more specific. So in addition to the actual structure of the ontology, HPO has been used to create a data set called the HPOA, so the HPO annotations, whereby sort of OMIM IDs or orphanet IDs, and depending on the group that has done the work, either Monarch Initiative or the Human Phenotype Ontology team or the orphanet team, have been used to annotate this condition. So basically, we have a large set of annotations to conditions to roughly around 6,000 rare diseases. And a particular area in kind of in the last 10 years, and the reason why I'm mentioning is because it's part of also what we've done in terms of work. So is the fact that the ontology or the annotations enable us and the community and those that have worked on this for the last 10 years to develop algorithms that go beyond exact matching in phenotypes, as you can see here and in this example, where you have two patients and a condition in the middle, where an exact matching would mean all three of them having the exact sort of HPO terms, to semantic similarity or fuzzy matching, where in principle you're able to compare sort of microcephaly and macrocephaly, which two distinct terms, but assign a measure of similarity between them, which then can be used to compute various things. For example, in ranking genes that could be associated with a phenotype profile or conditions. So the reason why I mention is because the work that we've done is obviously focused on or uses of both the HPO and the annotations, and the in part for comparative purposes, semantic similarity metrics. And to go back to, I guess, where I started the story, I usually, whenever I talk about our work, I'm trying to put things in context. And the context I usually use is this particular journey, which is a very, very oversimplified, if I may say so, journey of a patient of with a disease, and in particular a child. And you can see there that we kind of what we try to capture is these large components that deal with the initial complex clinical presentation, when there are various things that are happening, understanding the presentation and trying to find a specialist, or finding others that have a similar presentation, to finding a diagnosis, and then getting into this area where it's either about a treatment, which is only about 3 to 5% of the rare diseases. Have a treatment or some sort of therapy or care, and that care then sort of expand itself in various areas, touching on things like, for example, peer support, financial support, health system support, and then later on understanding how this care and the treatment perhaps has an impact on other aspects of life. And in particular, because I guess the predominantly the category of population that we're looking at is children, the education of these children and the transition into adulthood. So our work is in principle focused in principle in this area, while we sort of recognize that perhaps later on, if we do get where we want to go, or others perhaps are willing to help and go in that direction, I guess that this association of phenotypes over time could also be used sort of early on. But in what the area where we kind of want to focus on is this area of care and understanding how we are able to use trajectories of phenotypes and their associations with interventions in principle, sort of to guide coordinated care and care management for patients. So the work that we've done was basically, as I said, to try to look at time, and in particular at the onset of phenotypes, and I guess to some extent to try to automate the creation of a natural history of disease. Because when we started this, we didn't quite know exactly if it's going to work, but also perhaps to kind of make things a bit more easier to validate. We as I said, we verticalized the problem by looking at a single condition. That condition that we chose was Noonan syndrome. One, because it's multisystemic. Two, because also it has in some cases, depending on the subtype, it may have an impact on or a connection to cancer as well. And we've developed this pipeline, and I'm going to go bit by bit through this pipeline, and try to use both manual curation as well as automated extraction and assembly of knowledge using large language models, At a point in time where I guess they were in principle sort of emerging rather than being as established as they are now. Now, the starting point of our work and it was intentional, so to take basically a source of information that is well known and accepted in the community as being sort of a trustworthy source of information, which is gene reviews. And then from there, basically, to go step by step into understanding what is the profile that comes out of that particular source of information, what is the profile of the condition from other places, and understanding how we can then factor in the time and the onset of the phenotypes in order to build gradually the natural history. Now, just to take a step back, one of the reasons why we're doing this is in principle because we started by looking at the various profiles that exist for Noonan syndrome in existing knowledge bases, and kind of try to compare them. And you can see here, I guess if you look at all the OMIM so basically the HPOA where OMIM is annotated. The OMIM ID is annotated for Noonan syndrome with HPO terms and then orphanet, and try to see how do they compare just using simple set-based Jacquard index, which is kind of the intersection of the terms versus over the union, but also using a normalized semantic similarity, which in this case the similarity metric was the the usual Resnick of information content based similarity metric. And to see to what extent do they match? And you can see just by looking at these numbers that in principle there's a let's say high degree of semantic similarity. So the terms are similar to each other, but not necessarily overlapping. Now, obviously, there are other aspects that are, I guess I'm glossing over here. The fact that OMIM does subtyping of Noonan syndrome, and in this case, we're looking at Noonan syndrome 1 when it comes to OMIM, in terms of while orphanet just has the broader concept of the condition. So this was one of the first, I guess, the first motivations behind our work, and really trying to say, okay, well, clearly there are some areas that in which the condition was curated in differently by different groups, and not covering time in the picture. So we started basically to say, okay, if we have the HPO annotations either from to OMIM as well as to orphanet, what happens if we throw in another sort of data source, another information source, which is gene reviews? And in principle, what happens if we start by manually curating it? So what we did was to take the description of the disease of the condition from gene reviews, and we manually curated it by looking at in green here, by looking at phenotypes, looking at in yellow the interventions that are associated with these phenotypes, and then I guess more importantly, at the onset, which unfortunately is in this kind of light pink, and associating phenotypes to onset and phenotypes to interventions and so on. Now, the focus of this particular work was basically only on the onset, but we actually, when we did this annotation and we ended up doing the annotation for the entire group of rhizopathies, not only Noonan syndrome, we actually did it across all the dimensions or multiple dimensions, including this area of interventions, which is kind of the step that we eventually want to get to. So the result of this manual annotation process, again, if we do the same comparison, you see that the overlap is even less than what was before. So just looking at the first half of the table, where we're comparing the manually curated HPO profile of Noonan against the existing sources, and you see that the Jacquard index, which is again the set-based intersection over union, is fairly low, while the semantic similarities in these sort of rather reasonably high. So again, I guess from a semantics perspective, the term were in the same area, but not quite there. So an additional step what we did then, as in the lower part of of this table was to say, okay, what happens if we take the LLMs, that are out there at that point in time, which was GPT-4o and Claude Sonnet, 3.5. In the meantime, obviously, there's 3.7. And we just asked them to generate a natural history for this particular condition. And we take the profile at this point just as a profile without time. And, we compare them against change. And yes, and it was intentional. Yes, we were aware of the fact that, this can, involve data leakage, because, most likely, sort of, these LLMs were trained or were using things like, for example, gene reviews to be trained on. So, but we're actually counting on that, right? And in saying, well, I mean, surely if there is data leakage, then the overlap should be high. And in reality, it's not quite. So again, just the set intersection, so just the Jacquardines is actually quite low. And yes, again, the semantic similarity is good, but clearly, they have something else that they're capturing. So, and if we are looking at what exactly they're capturing, so by just using the top level abnormalities in HPO, you can see here sort of on the left is the curated knowledge sources, and you can clearly see the areas where things are different. So the GNAV is the one that we manually created, and you can see some spikes here in the immune system, the neoplasm, and the nervous system that are not well covered in other sources, as opposed to what we've manually curated, doesn't cover as well things like the head and neck, the musculoskeletal, and the cardiovascular system. And similarly, sort of, if we look now on the right, sort of the profiles that were generated by the LLMs, compared to the one that we've manually curated, you can clearly see that there are areas that they excel. Things like, for example, the metabolism and growth and the digestive system, which were, pretty much kind of rather non-existent in what we've curated, while in other cases that they seem to be fairly similar. Bottom line, the idea here is that we respected the fact and we acknowledge the fact that obviously, if you look at various sources, they might as well obviously capture various angles of that condition, which is great. But then the question is, okay, how do we go about sort of, creating a natural history of this disease by adding time and also trying to consolidate this knowledge? I mean, here kind of went into, I wanted to go into a bit more details by actually looking also at the time in where it's captured, because as I said, so when it comes to the LLMs, we actually asked for the natural history of disease in the various stages, from prenatal to late, which late basically is over 60s. And it's clear. One very interesting thing that's worth mentioning here is that if you look particularly at these two charts here, which I understand are not particularly readable, what I wanted to see is that the fact that in this particular case there's no brown, and there's barely any brown in these two. So basically, what I'm trying to get to is the fact that pretty much after the adult stage, which is, after 25 years, we actually didn't have any HPO terms describing the disease, while there were HPO terms associated to the disease by the models, which was one of the kind of interesting findings. And I'll kind of come back to that in later. I should have mentioned from the very beginning, so this is all published. I'm going to put there's a QR code at the end or in a paper where you can actually get the figures in a better quality than I'm showing them here. Now, we did this manual curation, but obviously the idea is that you can't actually do this across 10,000 diseases. Neither you can do it more, across 1,000 diseases, right? And not to mention that obviously it's a single source. So then we looked at understanding, how we can actually automate this. And the way we've done it was basically to take, like everyone else, sort of, PubMed, and we started by focusing just on looking at the abstracts that were about or mention Noonan syndrome. Now, just mentioning a particular condition, we all know that doesn't necessarily mean that the abstract or the particular paper is about the condition. So the first area where we used the LLM for as an agent was to check the relevancy of the abstract. So we actually used GPT 4.0 to check to what extent those particular abstracts, which are about 3,000. I'm going to go back to the numbers in a second. were relevant to Noonan syndrome. And then once we have those filtered down to a number that again, or a set that would seem to be relevant, we then took the phenotypes that was manually curated from gene reviews, and then we plugged them in and tried to understand to what extent they're actually present in those abstracts and in the correct context. So not just present and again being listed, for example, in a differential diagnosis context, but to be present and be associated with Noonan syndrome. And for this, again, we used GPT 4.0. And not because we're not trustful, we don't trust the model, but because we thought it's, always good to kind of have a, to have something, someone to double check the work. We introduced the second validation step, which was basically to validate the output of the previous validation step to see to what extent it was actually correct. Right. So while the previous one was checking to what extent so the phenotypes are mentioned in the right context and in the abstract, then we used Claude 3.5, which was was basically a different model to check if the previous step was performed correctly by GPT. The reason why I guess it seems a bit more convoluted is because and I guess we added a couple of perhaps extra steps was really to get to a point where this can actually be done fully automated and without having to rely on manual verification or manual validation. So we kind of try to do automated validation as well in the process. And as the last step, we, on the very last set of abstracts that was refined in this process, we did this phenotype onset extraction. So basically, for each phenotype that we found, we try to find an onset associated with it according to the, I think, 9, 12, 11, I think, categories that are listed in HPO. And the way we did this was by basically stop pairing up in a voting mechanism the both models and assuming that none of them has veto rights. So basically, if a phenotype and a pair of phenotype answer was found in an abstract, both models had to agree that it was actually there and was in a correct context for us to consider it in the final model. So this is the pipeline. This is the pipeline in numbers. So we started with about 3,000 abstracts, and we basically filtered down from 3,000 in the last step to about 1,400. You can see we started from about 20 to 200 phenotypes in the manually curated, then we ended up with about 152 after the automatic stuff validation. And then once we also added the onset to them, so to actually have phenotypes with onset paired up, we ended up with about 74 HPO terms. Now, on this side here, you see the natural histories generated by the two models, which initially started with fairly high numbers, as you can see. But then when we put them through the same process, we ended up with about half of the phenotypes. And this comes back to what I mentioned before about that brown missing, the brown color missing from the last two charts at the bottom, which is for pretty much for all the phenotypes that were listed in these generated natural histories. After the adult stage of the disease, we found absolutely no scientific evidence. So there was no abstracts. And I understand, I'm going to come back to this in a second. Yes, it's abstracts, not full texts. We found no abstracts that mention that phenotype in that context. And in particular, in most cases, they were basically neurodevelopment and neurological stuff in the phenotypes, and they were not mentioned in that context in scientific literature. So this is kind of the validation when we kind of you can look at the manual versus the automated and evidence-based validation that we've done. So if you look at basically the manual curation, we ended up with phenotypes across the entire spectrum of the of onset from prenatal to late. And you can see there are barely any phenotypes in this late adult stage. The automated step, unfortunately, I skipped over it, but we actually try to do automated extraction of phenotypes from gene reviews as well, just to see to what extent stuff we actually need the manual step. And you can see that there is some loss. And in the paper, we actually discuss exactly what's the reason why it lost. There were some cases, some phenotypes that were missing stuff synonyms. And we know that the concept recognition libraries that we use, they couldn't find them. So there is some loss, but not that much. Versus sort of, if you look at the evidence base, which basically means, actually finding finding the phenotype with onset and scientific or PMID stuff attached to it. You see the numbers, that are pretty much kind of in concordance, both in the manual extraction perspective as well as the automated extraction. Now, this last step again, it's pretty interesting because it's an area that we were kind of wondering to what extent is an artifact of the fact that there were actually no studies, on Noonan syndrome, in the later stages of life, or perhaps the fact that, as I mentioned, we did use abstracts. Indeed, they were not full texts. Perhaps it was a limitation, which was a limitation also of our study, and the fact that we were not covering enough material. So the result looks a bit like this. When you look at the when you look at again, the brown is the gene reviews, gene review based the manual one, and then the areas that are covered across the 11 onsets. And then the rest of them are based on study labs. And in particular, you can see again there are differences, and in some cases there are quite marked differences in terms of the areas of the HPO terms that are being covered, which then led to a final question, which is very similar to the initial question that I asked on this picture, which is, if we have these, what do we do in terms of consolidating them? Because clearly, kind of what we were able to find evidence for in the literature, covers certain areas, but clearly it's not comprehensive. And while some could claim that perhaps the phenotypes that are being produced by the LLMs are hallucinations, they could very well be. There is a question of whether those hallucinations that we could claim to be hallucinations or assume that they could be hallucinations are actually not hallucinations, but rather us not understanding where the data is coming from, because we don't understand what data was ingested in the first place by the models to produce these natural histories. So the last step that we've done, and then I'm gonna wrap up because I'm already over time, is basically to propose. We proposed a couple of ways to consolidate this knowledge. Kind of the knowledge that comes that was manually curated, the knowledge that was automatically extracted with scientific evidence, and then the knowledge that one could actually get by automatically from LLMs just by asking for it. And in principle, the consolidation strategies were around basically making a decision with respect to whether you want a model in the end that is fully grounded in scientific evidence or not. And it's all basically, at the end of the day, it's up to everyone kind of that wants to use the output or the outcomes of this work in a particular context. So, but to take a step back, once we had this kind of this natural history map. I didn't put the picture because it's like very, very small and a lot of phenotypes, across kind of the 11 stages. So I put this one, which is it's a bit easier to understand. We went back to the EHR here in KKH in Singapore, and we looked at patients that were treated and were diagnosed with Noonan syndrome within the hospital. And we went back into their history and the medical history in the AHR, and we tried to say, can we validate the phenotypes that we find in the EHR across time against what we generated by putting together kind of these phenotypes or associating the phenotypes with onsets, sort of, from kind of this automated pipeline. So we had only 6 patients, unfortunately, and this is an example that I show you here, where you can see that the patient was first seen in a juvenile stage, but then sort of, and it continues to, for a number of years to be seen there. The everything that you see in bold across the various anatomical systems. So basically, each of these bubbles is one of those top-level abnormalities in HPO, but just depicted in a graphical manner. Everything that's in bold was actually mapped um, against what we found or what we built in an automated manner at the right time. So kind of things like, for example, motor delay and wide intermammillary rest distance, were mapped exactly where we found them or in the automated natural history of the disease. Now, there are some others that were not quite exactly there. So things like, for example, short neck wasn't quite there. There was a term in what we found. It was in principle, one level higher or one level. In this case, was I think one level higher in the hierarchy of HPO. So very close enough. And there were some like syncope or wide nasal bridge there was at least a sibling of this or direct sibling of this term in what we found. So in principle, in this particular case, and in reality, stuff, in all 6 patients, we were able to a large degree to validate what we've done or what we created in terms of this mapping of phenotypes across the onsets and across time. But interesting enough, we also found, so by doing this exercise bottom up, we also actually found phenotypes that were not documented at all, at least in neither by us, neither what we found in the literature, which then raised the question whether those, they were secondary phenotypes or actual phenotypes of the disease. So basically, in a nutshell, because I think that was basically it. So in a nutshell, what we tried to do was to start by looking at a single condition, which was the Noonan syndrome, and to try to understand to what extent we can capture not just the phenotypes of the condition, but the phenotypes with their appropriate onsets, and across basically the life course. As a precursory step to what we want actually to be able to do eventually, which is not just have these phenotypes each in each of the onsets, but also to understand to what extent these phenotypes have some sort of relationship across time, and where exactly. So the interventions that I showed you in the manually created example from gene reviews, where those interventions come in, sort of in each of these onsets, and later on. So what is the impact of both these phenotypes, the relationship, as well as the interventions across other dimensions of life, and in particular on the education of the children. I'm very grateful to all the colleagues that have contributed to this work. Now, I did single out here Gareth and Somia, who are critical to making this work happen. But nothing would have happened without the work done by the HPO team and by the Monarch Initiative team. And thank you so much. Happy to take questions.